Hello second year students, we are starting with the bacteriological analysis of milk. So very firstly we are going to write down here the name of the practical. Okay, so we will write like this: milk analysis. Here analysis of milk is done with the help of certain specific media. First of all we have an agar plate. Okay, an agar plate is colorless and it will not have any color because here only agar, peptone, yeast extract and NaCl are added. Now this is the plate in which we have added dye, indicator dye. This is McConkey's agar plate. Okay, so one by one we will write down the name of media and then we will try to understand. This is our nutrient agar plate, that is N agar plate. Okay, so we will write like this: N agar plate. Okay, so this is our N agar plate. Now the another one is McConkey's agar plate. So we will write like here, it is pink in color because of the color of the indicator dye. So you can see very nicely that this is McConkey's agar plate. Okay, this is McConkey's agar plate. Then after we have another media with us, and that is EMB agar plate. You can see that color of EMB agar plate is mauve color, or you can say that it is in between the shades of purple and pink. So this is our EMB agar plate, that is eosin methylene blue agar plate. So these are the plates which we are going to use today. Okay, 3 different plates are to be used, and then after the biochemical tests are to be done. Now in case of biochemical tests, we are going to perform 3 different biochemical tests. In first we have prepared 1% peptone, which is colorless or slightly yellowish. This is our GPB tube, that is glucose phosphate broth, and this is our citrate slant, which is already having parrot green color. Okay, so here we will write like this. Here very firstly 1% peptone. Peptone. This way we are writing 1% peptone. Then after we are writing here GPB broth, glucose phosphate broth, GPB glucose phosphate broth, and last one is our citrate slant, Siemens citrate slant. Okay, citrate slant. So you can see all the biochemical media very nicely. Okay, this is our citrate slant. So now what is to be done? On the first day we are going to inoculate 2 different media plates, that is N agar and McConkey. Now how to be inoculated, so that we will see now. Okay, so you have to imagine that the pen which I am using is our nichrome wire loop. Okay, first of all we will make the sectors 1, 2 and 3, 1, 2 and 3. This is our first sector, this is our second sector, and this is our third sector. Okay, so you can see very nicely that I have prepared 3 different sectors in case of an agar plate. Now the same we have to do in case of McConkey's agar plate. Sector number 1, sector number 2 and 3. So we will also write it down with the help of glass marker on the bottom of the tube like this, bottom of the plate like this, 1, 2 and 3. Okay, so after doing this much of the work, now we will use our milk sample. Milk sample is to be taken from any dairy, or it can be also taken from the you can say the pouch of any okay, well known dairy like Amul or something, okay, or you can also take pasteurized milk. So any milk will be available, whatever is available can be used for this practical. Now from the tube of milk sample we have to dip the nichrome wire once inside, and then after you have to imagine that this pencil I am having is our loop. Okay, so now how I am going to work with the first sector. Now this is to be done in between two different burners which are already lighted on. Okay, so aseptically now we are starting the inoculation in the first sector. Okay, so it has to be done like this. It has to be done like this. Okay, so the first sector is to be inoculated now very nicely. Okay, so you can see that I have started inoculating. Okay, this is my nichrome wire. Very slowly and steadily by covering the entire sector. Okay, like this. This way we have to inoculate the agar plate. Okay, so we have completed sticking first sector. Now we will go for another sector, sector number 2. So for that we will make the change, and we will move the loop, or you can also move the plate, and now we are going to inoculate the sector number 2. So with the help of loop again, like this. Okay, very steadily, and you have to make the stalks very close to one another. Okay, it is to be done like this. So our second sector is already inoculated. Now we will go for inoculation of third sector. So that is to be done. Okay, very nicely. Now this is our nichrome wire, and we have started sticking the third sector. So by taking all precautions that it is not touching to the another sector, we are inoculating the plate. Okay, like this. If you may make very close sectors or very close stalks, it will give you more good isolation of colonies of bacteria. You are able to dilute it out very nicely when you make the very close stalks. So our agar plate has already been inoculated. Now we will go for McConkey's agar inoculation. Now in case of McConkey's agar plate, again we will start with the sector number 1. Okay, first of all we have to dip the loop in suspension, and then after we are now with the help of loop starting the inoculation in first sector like this. Okay,